We live on the cutting edge of physics, materials science, engineering, and nanotechnology. Because at Lockheed Martin, we're engineering a better tomorrow.
Good morning, and thank you so much. As was mentioned, my name is Sara Volz. I'm a student at MIT, Massachusetts Institute of Technology in Boston, and I'm here to talk to you about basically what my journey has been like as a student researcher. So, see how the next version works. Oh, look, it works. So my work in high school was on algae biofuels. So you're all probably aware with the energy crisis as it kind of stands today, and we just heard a little bit about how that's impacting ecosystems, but what you may not know is that pond scum can actually be turned into petrol. And actually, as it happens, any sort of natural oil can be converted into biodiesel, which is an alternative fuel that can go straight into a diesel engine. And chemically, it's very similar. It'll work great, and you don't have to change much about existing infrastructure. In fact, you can even make these fuels out of the vegetable oil that you find in your kitchen. So why do we care about algae specifically? Well, the simple reason is that algae are actually a lot more effective at producing these types of oils than other crops. Specifically, the problem with land-based crops is that, well, if you're growing soybeans to make fuel, you are not using those soybeans for food. And so even if you used the entire land mass of the planet, that would not be enough to fuel the US economy if you were just using that to grow soybeans, turn that into oil, and then turn that into fuel. So that's where algae comes in. The real problem with algae, as it is right now, is that it's actually not quite economically feasible to compete with fossil fuels, with diesel fuels, and with gasoline. And that's where my work comes in. I'm looking at trying to improve algae oil content to try and make algae biofuels more feasible. So in general, the idea behind this is you've got algae taking in carbon dioxide, like plants, that's what they breathe. They take in carbon dioxide through photosynthesis, turn that into a carbon source, and they use that to make all sorts of molecules that they need to live and survive. And one class of those molecules are oil molecules, or lipids. And these oils are the same ones that you would use to make fuel. But this is a very complicated series of reactions going from carbon dioxide to these oils. And in general, if you're trying to improve the production of these oils, you'd want to change one of those very complicated mechanisms on this whole path. The problem with that is there's a lot of regulation, there's a lot of enzymes and proteins that are involved in this process, and it's very complex. So if you want to change something about it, say change an environmental stimulus and try and get it to produce more oils in sort of a temporary setting, or change something genetically, try and overexpress a gene, those approaches might not always work. So my idea was to do something slightly different. And at first, this might seem a little counterintuitive, but I actually read about this herbicide, a chemical that actually blocks the algae oil metabolism. It's used to kill weeds because it prevents plants and algae from producing these oils that it needs to survive. And at first, this seems counterintuitive because why would something that blocks oil content help to improve oil content, right? But I had the idea to instead use this chemical to force the algae to produce more oil through a process called artificial selection. The basic way this herbicide selection process worked is the goal is to increase the enzyme activity of this particular factor that's involved in the algae oil metabolism. So in general, in a large population of algae cells, most of them have normal levels of oil, but there might be a couple of variant cells with unusually high enzyme activity and unusually high oil content, and usually these are nigh invisible in the population. They're just very rare, so you don't really see those effects that they have. However, if you change the environment that these cells are living in by adding this herbicide that prevents cells from surviving if they don't have enough oil being produced already, then you are basically making an environment where suddenly that rare variant cell is extremely successful when compared to the cells living around it. So you just kill basically all of the cells around these variant cells with unusually high oil content, and in the end you're going to see a population shift. At least that's the theory. Basically, these cells that have been growing in this herbicide environment need to have artificially increased their oil production in some way in order to survive these harsh conditions. And that was the idea behind my work in high school. I basically wanted to see if I could make this herbicide-tolerant population and see if it had this high oil content. And it took me a couple of years, but here's like the takeaway results. I was able to get much higher oil content in my herbicide-treated cells. That's that yellow and orange bar next to the blue bar, which is the untreated cultures. Basically, I was able to show that after a long period of this herbicide treatment, I could take it out of this treatment, and it would still have increased tolerance to the herbicide and increased oil content. So the implication would be that I've genetically changed something in my cells. Basically, via evolution, these cells have become more tolerant to conditions that prevent them from making oil, so they're better oil producers and therefore would be a better source of biofuel. So this was a very exciting result to get, and it took me a couple of years to get it. But right now I'd like to show you how I got that result, basically the location. This was me in high school. As you can see, my hair has changed, but basically this is the lab setup that I made under my loft bed. You can kind of see the slats at the top of that picture. Of course, like any good laboratory, it's got the requisite bubbling jars of goo. I've got a microscope that I got for Christmas. I've got flasks that I begged, borrowed, and stole from some scientists. I've even got like a really dinky old centrifuge that I was able to get used from someone on eBay. And yeah, there's all sorts of things on the internet. You never know. And I sort of built this laboratory up over the span of a couple of years in high school, but I never thought it would get to the point that it did. And so I'll show you sort of what my projects used to look like. So this was me in 6th grade. I really liked neon. So that's my first science fair that I ever went to, my first regional science fair that I ever went to. And I was doing a project on on whether or not cat saliva had antimicrobial properties. As it turns out, probably should just use Neosporin on your cuts. Who knew? But that first taste of science fair in 6th grade was what got me really interested in continuing to pursue scientific inquiry, basically. I had always liked science, but I'd always liked a lot of things. I'd been interested in history, I'd been interested in writing, in poetry, in theater, but after that first taste of science fair, I really realized that science was something that I could do even in 6th grade. And in 7th grade and 8th grade, I really got into using biodiesel for my projects. So I was doing various chemical reactions with oil to try and see if I could increase the efficiency of these chemical processes. I also got to go to the state science fair and where I'm originally from, which is Colorado. And the experience of being at that fair and seeing what people my age were able to create and produce was, there's really not another word for it other than transformative. I was able to look at the projects that people like me were doing, people my age were doing, and they were changing the face of scientific inquiry. They were making real, meaningful discoveries that were published sometimes, that were patented sometimes, and that were really making an impact in terms of scientific innovation. And that's when I knew that science fair wouldn't be something that would let me go. And in high school, in 9th grade, I got into Algae. I also got to go to the International Science Fair for the first time. It's sponsored by Intel. It's a huge event, 1,700 students, 75 different regions and countries, over 4 million in various cash and scholarship prizes. That experience was amazing because I got to meet people not just from all over my state, not just from all over my country, but from all over the globe. And I continued those experiences. I got to go to several different international fairs in high school. I got to have Facebook friends from China and Egypt. I got to see people from Russia who were doing work with biodiesel. And it was amazing to me that that science could be something that brought together people from all across the globe. So here is a picture of about half of the exhibit hall at the International Science and Engineering Fair. And any of you could be at any of those booths. The amazing thing about this scientific inquiry is that we're all capable of it. All you need is a project and an idea. When I first got into science fair in 6th grade, I really didn't have much hope in terms of what I'd be able to accomplish, because I thought that, well, I mean, all the people with really good projects work in, government labs, or they find some sort of professor as a mentor, and people certainly do that. But I really want to drive it home that you don't need to do those things. If you have an idea, then you have all the resources you need. All of that can come from you. You can go out and you can ask people for advice. I emailed tens of professors, sometimes wouldn't get responses, but on the occasions that I did get responses, it was really a wonder to be able to follow up and realize that, yes, so some of these experiments, sure, I'd need some fancy equipment for, but others of these experiments, you don't need much to grow algae, for instance, other than a nice seawater medium and a flask and a microscope, maybe, if you want to look at how the cells are doing. For more complicated analyses, I was able to access some facilities at research institutions after I basically spent a lot of time, asking for help and asking for access. But those were not the things I relied on because, for instance, one of the main institutions that I worked with was 3 hours from my house, so I really could not rely on that very much. But what this all eventually led to was, so I won the Intel Science Talent Search in 2013, and what this really meant for me was all of these experiences with science fair really put me in the position that I'm in today, which is at MIT and able to be pursuing groundbreaking research with some incredible professors and some incredible minds. And so I want to talk about that a little bit. The work that I'm currently doing is very exciting. It's on genome editing. So what is genome editing, first of all? So as you may know, our genome is basically the set of instructions in our cells that encodes everything about us. So the basic theory behind biochemistry is you've got DNA, so your genes. Those encode another molecule called RNA, which is similar to DNA in structure, but it's only one strand instead of two, and it's got some chemical differences. Then that RNA will be translated into a protein. And proteins are made of amino acids, so they've got a different structure. And there's tons and tons of proteins in your cells. Those are the main movers and shakers behind the functions that your cells carry out. And proteins have a huge diversity of function. So if you want to be able to change things in your cells, you want to be able to change proteins. And if you want to be able to change proteins, what you really want to do is go back and change the genes that encode those proteins so that you can change permanently the instructions that are dictating how that protein functions, so that you can basically have a cell that not only is altered, but will go on to make daughter cells that are also altered. So there's basically 3 steps behind genome editing. First of all, you need to find the gene that you want to edit. The human genome, for instance, is something like 3 billion base pairs long, so a base pair being each of those little letters of instructions in your genes. So obviously there are tons of genes, and you need to be able to find the right one that you want to alter. Second is you want to cut that gene. And it turns out you want to cut that gene because that is how biochemically you'll be able to change it. The third step is you insert or delete or change that gene. It sounds all very simple, right? Well, so let's talk about step 2, which is cutting DNA. It turns out there is a type of protein called a nuclease, and nucleases are designed explicitly for that purpose of cutting DNA, just molecular scissors, if you will, that cut it right in half. So that's taken care of. Once we get to a spot in the DNA, we can cut it, for sure. So let's talk about step 3. Once those cut ends of DNA are available, you can insert or delete or change a gene. Deleting is probably the easiest, messing up a gene, because it turns out that there is a lot of machinery in your cells that is committed to changing how your cells, how your DNA works. So DNA in your cells gets damaged all the time, and strand breaks like this do happen, and your cells need a way to fix those. So the committed machinery that your cells have for fixing it often just kind of glue the DNA ends back together in a manner that's pretty messy, and that will usually ruin a gene. It'll cause a deletion or an insertion that basically messes up everything about the instructions that make up that protein. So you can delete a gene that way. If you want to insert or change a gene, that's a little bit more complicated, but not too much more complicated. For this process, you actually add in a piece of template DNA that you, the researcher, designed that encodes a new set of instructions. You just make sure it matches on either side of the place that you're trying to change, and the machinery in your cells will recognize that it matches and will repair that piece of cut DNA based on the template that you introduced. So all of this is sounding pretty good so far. We've got a way to cut the DNA, we've got ways to change the DNA once it's been cut. So what's the problem? You'll notice I haven't mentioned step 1 yet, how to find the piece of DNA that you're looking for. And it turns out that in the field of genome editing, this was definitely the hardest part for a very long time. Because the only ways that we knew how to find a particular site on the DNA was using proteins. And proteins, the examples that I have on this slide are zinc finger nucleases and Tal effector nucleases, the TALEs. Those are basically just different types of proteins that recognize specific DNA patterns. So if you've got a DNA pattern that looks a certain way, you can use one of these nucleases to target it. And you can redesign these nucleases to a certain extent. You can change these proteins so that they recognize different patterns of DNA, but that's a very difficult process. It's costly, it's time-consuming, and it doesn't always work. And so that's what has kept this mechanism of genome editing from gaining widespread traction in the scientific community for a long time. The fact that we just couldn't design these nucleases well enough. A couple of years ago, though, that changed when we found a solution. Some scientists at MIT and some scientists at Berkeley were able to discover that there is a special type of nuclease called Cas9, and it's associated with this whole really cool system in bacterial cells. It's actually a system it's specifically a system that is designed for finding certain patterns of DNA and chopping them up, and that's perfectly what we're looking for. And this nuclease actually uses an RNA molecule to find a DNA template to cut, which is perfect because RNA, like I mentioned a little bit earlier, is very chemically similar to DNA. RNA is written straight from the template that DNA provides via the same sort of base pairing that DNA has. So that means that if you've got an RNA molecule that has like 20 base pairs, has 20 letters in its sequence, you know exactly where on the DNA that's going to target. And so because Cas9, this nuclease that cuts DNA, uses this RNA in order to target the place that it cuts, now we have a perfect way to find places in the genome to cut. So with this system, and you'll definitely hear it in the future, and you might have heard about it now, it's called CRISPR-Cas9. With this system, the face of genome engineering and of biochemistry is radically changing because now we have the ability to go in, cut genes, take them out, put in new ones. This means that we can research how genes function more easily. We can maybe make like different gene products. So maybe you want to produce a lot of a particular protein. You can do that a lot more easily. Or say you have some sort of genetic disease. You can cure those diseases with these techniques using gene therapy. And I'd like to show you a picture of what Cas9 actually looks like because I think that's really cool that you can see the molecular structure of proteins and of these biological molecules. Too often people just kind of draw lumps on the screen, which I've been doing. But in blue is the actual Cas9 nuclease, in green is the DNA, and in red is the RNA. So you can see that the nuclease grabs onto the RNA in red, and that RNA recognizes the DNA, and then this nuclease will cut that DNA. It's a really cool system. So to quickly mention my work specifically, I have been working on making Cas9 into something that you can turn on and off. And basically I've done that by splitting it into two pieces so the protein is inactive, and then making it so those two pieces will only come back together in the presence of a small drug molecule. So that means you put in the protein, you put in the RNA that targets the gene you want to change, and it'll kind of just sit there in the cell until you add the drug molecule, and then bam, it turns on, the two pieces come together and edit DNA. And what's so cool about this is that in 6th grade, even when I knew that I was interested in science, I didn't really have this vision of going forward and getting to do research, and I didn't know what that would actually look like. Like, you have to be in your 30s, right, to be making actually groundbreaking changes, right? But what I found out is that that's not the case. When you're 12, you can make interesting discoveries. When you're 16, you can make interesting discoveries. And when you're 20, like I am, and in college, then you've got so much access, so much more access than you have when you're younger. But it's really the experience, the journey from 12 to 20 that made me realize that I want to do research, and not only that, but I could do research and could make all of these discoveries. I certainly wouldn't be in this lab and wouldn't be doing the work I'm doing now if I didn't have the experiences with science fair that I had. So I want to encourage all of you to, if you're interested in research, to just ask a question. And I want to encourage you to, if you've got a question about science, if you've got something that you want to change, if you've got a project that you want to do, to be that change that you want to see in the world, to be the scientist that you wish you could be, because you don't have to wait on that. You can do that now. So thank you so much. I'd love to take questions.